record and you can go ahead whenever you're ready. You have 10 minutes. I'm going to put the timer on.
Okay.
Let me know whenever you're ready.
Yeah, I will. Just a second.
Okay.
Mm, Just a second. I'm having a little problem. I don't know why.
That's all right. Yeah, take your time.
Do I have to share my screen and show the poster and then talk?
Yeah, you can do that. You can share your screen. You should have the ability to share your screen and then we'll be able to see you as well.
Are you able to see my poster?
Yes, I'm able to see it. So I can see it. So let me know whenever you're ready and I'll start the timer.
Yeah, I'm ready now.
Okay.
Yeah, good afternoon, everyone. This is Tejasvi, a biomedical and health informatics graduate student. I'm currently interning at a company called Health Compass as a health informatics and data analysis intern. So we are currently working on a project called Vital 110, where we are trying to build an AI model to know the future risks in prayer. So today the healthcare system has become reactive. We only go to the doctor only when we see the problems. So we are trying to change that because we have done a lot of research on scientific evidence and we gathered some valuable information. Like in the scientific evidence, you can see that a 2020 umbrella meta-analysis in the British Journal of Nutrition, which was done on 4.8 million participants, showed that healthy diets lowered the risk of type 2 diabetes by 19%, colorectal cancer by 16%, while poor diets raised diabetes risk by 21% and the metabolic syndrome by 26%. Along with that, another significant study is a 20-year-long longitudinal study by the NIH. It involved 7,000 participants, and it also concluded that people who slept less than 6 hours per night had 50% higher risk of developing type 2 diabetes and 40% higher risk of obesity, highlighting the importance of sufficient sleep for metabolic health. So these are just two examples of multiple articles which we taken into consideration. So in our approach, we identified 8 key behavioral risk areas, that is poor stress management, unhealthy diet, physical inactivity, insufficient sleep, lack of preventive screenings, inadequate adherence to medical care, smoking, and substance abuse. So we are trying to educate our patients by telling that if you are trying to sleep less than 4 hours for 20 days, you are at the higher risk of developing cardiovascular disease by 3% in the next coming 10 years. So this is the model which we are trying to develop. So our approach is to create a personalized health questionnaire. So which tries to act as a bridge between what we know and what we want to know. So it is backed by scientific evidence. It is one of its kind evidence-based tool. So after a person joins our insurance program, we will make them answer this questionnaire. After that, we are going to grade the patients into low risk, moderate risk, and high risk. After that, we also have collaborated with a lot of medical stores and wellness programs. They're going to guide us through healthy daily habits because we also believe small daily habits are the most critical determinants in future health risks. So, and this questionnaire combined with AI models like random forest and logistic regression, analyze behavioral patterns, and identify early health risks of chronic diseases. So we are trying to change the motto prevention is better than cure by telling that it's only better than cure only if we are aware of what risks we are preventing and at what risks our body is at the verge of acquiring. And in the poster, you can see this is how our app is going to look. This is not the exact app because we are still working on it. This is like a small example. We have taken this picture from Google. And in the current phase, we are currently in the data collection phase. As I said, we have 10,000 people in our health insurance program. So we are taking them into consideration and making them curate our questionnaire. So now these users are voluntarily sharing lifestyle and behavioral data through our app's dynamic questionnaire system. This real-time input is actively feeding into our AI models, allowing us to validate risk predictors and fine-tune performance across diverse cohorts. So in the future, we are trying to create an app with interface which tells patients to change their daily behaviors to prevent future risks. Yeah, this is it briefly.
So, okay, some questions for you. Like, do you have so what's the plan? Because um, you have done some work. You're planning to analyze data, but the data is not available. So, what are you planning to do?
So for now, we are still in the data collecting phase, but we are trying to check our model with already existing data. We are trying to, we have a nerds called AI, we have AI nerds called Victoria. Right now, we are training that nerds with all the questionnaire. And then we have a data team which is working on AI models. We are trying with the existing data as of now to see if our models are working precisely so that we can use the real-time data.
Yeah, but you said you don't have the data yet.
Yes.
So what's the plan to test your system without the data, possibly?
We are trying to know the data format and use the existing data, professor, because we are the first wellness program who are trying to include preventive analytics in an app, but we already have studies which they have used a lot of data to know the risks, to prevent the trends, to know the trends. So we are trying to use the data. As an intern, I'm trying to use that data to know.
Okay, so you do have some data available. Okay. Okay. Well, thank you. Is there any other question? No? Well, thank you.
Thank you, professor.
Thank you. All right, so I will move on to the next person. It doesn't look like Raul is here, so I will move on to the next person on the list. 1 second. Okay, it looks like Sravani is not here. Okay, so okay, Manny Deep. Oh, Manny Deep is not. Yeah, Manny Deep, I'm going to spotlight you. You're the next one to go.
Hello, hi.
All right.
Hello, hi. Yeah,
1 second. I'm going to set up the timer really quick, and then you will let me know whenever you're ready, okay?
yeah, okay. I'll see you.
Okay, the timer is ready. You let me know.
Yeah, sure. Can you guys able to see my screen?
I can see your screen.
Yeah, sure. I'm ready actually.
Okay. Is it possible to put it in presentation mode, please?
Yeah, sure.
Or to expand it? Do you think you can put it, I don't know, if the arrows that are next to the printing button,
Yeah, sure.
is it possible to expand it that way? Just so that we can see your poster a little bit clearer.
Okay. Yeah, sure. Did you get a?
Yeah, maybe the one, I'm not sure, but maybe the one next to the cog wheel, next to the little settings button at the top right-hand corner. I
Yeah,
don't know if you can expand that. I'm not sure, but maybe if you could try that. Yeah, that's definitely a lot better. Yeah. I don't know if anybody else can see that better now, but all right,
yeah,
you let me know whenever you're ready.
yeah. I'm right now ready, Ali. Yeah,
All right, you're good to go.
yeah. Hello, my name is Mandeep Reddy Potula. Today, I want to take you on a short but important journey on breast cancer. This is not just a health issue; it affects people everywhere, no matter where they live and who they are. They are affected with the breast cancer. And coming to my topic, my mentor is Dr. Isabel Pichandaris on breast cancer. Introduction is the study of this study completely explores on breast cancer survival prediction using both traditional and statistical methods and also machine learning techniques. I have used like both techniques in this. One is traditional techniques also consist of like Kaplan-Meier and some survival methods, and using some machine learning methods are random forest methods and from um, gradient boosting methods I have used in this. Coming to these methods, the study begins with data cleaning and preprocessing of TCGA breast cancer dataset, including feature selection, encoding, and handling missing data values, and exploring data analysis. EDA was performed in Kaplan-Meier, covers the visualizations to understand feature distributions and survival methods. Traditional survival analysis involved Kaplan-Meier stratification and the Cox proportional hazards in identify significant and material factors. Machine learning models, random forest method, and gradient boosting and fast kernel SVM were applied in this traditional technique methods. And data was standard and survival data was structured using SCID survival formats. Model performance was evaluated using the concordance index and accuracy for classification-based approaches. My project main aim is to test different models like Kaplan-Meier, Cox PH, random forest survivals in breast cancer patients, and cleaning the data, missing values, and also finding the duplicates, censored cases to make it ready for accurate survival analysis. We measure how well it models work using the concordance index and study survival difference by using race, age, and weight. The objective for the project is to learn and apply both traditional methods like Kaplan-Meier and Cox-PH and modern machine learning models for predicting breast cancer surveillance. To evaluate and compare these models using performance measures like the concordance index and helping support better data-driven clinical decisions. And the project mainly focuses on improving predicting patient accuracy using clinical and genetic data from TCGA dataset. We evaluate models using concordance index, and this helps us more assess which approaches best personalized data in it. My future works are like I'm planning to use a large data set and real data in it, and adding medical images and genetic data can help improve the prediction value more. And in future, these data models can be used in hospitals to help doctors plan treatments and predict outcomes in an easy way in a real-time data. And coming to the results, these machine learning models were evaluated to perform survival analysis of breast cancer using both clinical and genetical data, and the gradient boost technique was used more like in this project. It showed the good accuracy 0.9534 and 0.9798 using in the random forest machine learning method. The gradient boosting technique model was used very well. Coming to the traditional techniques, it didn't show too much accuracy in it, but fast kernel SVM also showed some predictive ability in C index 0.91333 to predict like what's the breast cancer tumor size and everything. The traditional Cox proportional hazards model has lowered C indexes of 0.7757. The traditional methods techniques, machine learning techniques results were shown very less. And coming to the discussion, machine learning models like random survival forest and gradient boosting perform better in predicting patient survival also. During this ability to model non-linear and complex relationships, and Cox proportional hazards remains valuable for this interpretability and clinical relevance, helping to identify important risk factors in it. Now, the project shows that no single model first all needs use a combination of methods offers both accuracy and understanding. This approach is scalable and can be adapted to other cancers or datasets for broader impact in personalized medicine. Conclusion for this project is combined traditional methods and machine learning methods are used to predict breast cancer's survival using TCGA dataset. Machine learning models like random survival forest and gradient boosting showed higher accuracy in traditional methods also. Cox model gave useful clinical insightful but had lower predictive power. The pipeline successful handled missing data and also sensor records and duplicate values has been identified in this. Results highlights the value of data-driven tools to support personalized treatment and improve patient accuracy outcomes.
All right. Well, thank you. I don't know if anybody has any questions. I'm going to start with questions, but feel free to ask questions as well. What I don't understand is you have not explained the figures that you have here. You have some figures. Can you explain to us what the figures mean? And then I see also a heat map.
Yeah. Here the model comparison for this like a grade in this picture, it's showing the gradient boost accuracy for this is for the model. I have built this model using this machine learning method. It's 0.97 accuracy. It's showing over here 979098, and random forest method using this machine learning models are created to predict better breast cancer using this patient outcomes. It's showing by using this random forest method, it's only shown like 0.9534. A slight difference, but the outcomes comes very fast, like for the using this gradient boosting techniques also. And here we can see the other technique, fast kernel SVM also. It's showing 0.913. And the traditional Cox proportional method, using this traditional techniques also, we might use like I have used in the methods like two different types, traditional and machine learning, right? The traditional technique is used for this. The outcome is very less. It's showing 0.7757 in this model. And the results are here, like heat map for the breast cancer. And here we can find that breast cancer data, how will be the Cox proportional and C index value is getting increased over here in this data. And in the correlation matrix, we can check it out, like what are the things we can do in the to predict the patient outcomes, and how the age and race by using this weight to support fair and personalized care for the patient.
Yeah. So can you be a little bit more detailed about which features you found associated with the survival? Have an association with the survival? Which
Yeah,
features in particular?
in features, basically using this survival rate based on age and race. Age is main concern here. Like some patients will be using like 30-35 years is not affecting more, and but a patient at the age of like 60 or 65 years has a low level of survival rate in this model, Professor.
Okay. Yeah. Because I mean, for breast cancer, probably gender is not important, right?
Oh yes,
Yeah.
yes, Professor. Yeah.
All right. Are there
Yeah.
any other questions? No? All right. Well, thank you.
Thank you. Thank you, Professor. Thank you for having me.
Thank you.
Yeah.
All right, I will move on to the next presenter. So it looks like I'm just going to go through one more time and see if there's anybody from before. So Raul is not here. Okay, Sravani, I don't see. Okay. Please correct me if I mispronounce. Is Miriam here? Miriam? Yeah, Miriam.
Hi, yes, I'm here.
Hi, would you? Okay, I'm going to, you looks like you're next on the list. I'm going to pin you. It looks like I know your camera's off. Do you mind? It looks a little blurry though. And I will prepare the timer.
Yeah, your camera looks blurred, and plus you want to share your screen as well.
Okay. Okay, sorry. I'm having an issue.
That's okay. Take your time. I do want you to know that your camera is blurred. I don't know if you just aren't intending to have your camera on. That's okay.
It's
Just
on.
let
It's
me
on.
know.
I'm not.
Okay.
That's okay. All
Um,
right. Let me know whenever you're ready. I have the 10-minute timer ready to go. So you let me know.
sorry, I'm just trying to figure this out. I don't know why it's not working.
No, that's okay. Can you at least share your screen so that we
Yeah.
can see the poster? That's the most important.
Okay. Okay. Can you guys see my screen?
Yes.
Okay, amazing. Can you guys see the poster?
Yes, we can.
Okay. Hi, everyone. My name is Miriam Rashid, and I'm a graduate student at SUNY Oswego in the Biomedical and Health Informatics program. Today, I'll be talking about enhancing patient-centered care through mobile health application. Well, first, we're going to start with the introduction. Mobile health or mHealth app help people manage their health through their phone. They let user track symptom, get medication reminder, message doctor, or even check test result. These tools become even more important during COVID when many people couldn't go to the doctor in person. But while mHealth app are useful, not all of them are easy to use or accessible for everyone. Some don't work or will with hospital system or other aren't made for people with disabilities. That's why I wanted to evaluate how well mHealth app support patient-centered care. In this project, I focused on four main things. First one, do these apps help patients talk to their doctor? Are they easy to use and do people keep using them? Can people with disability use them comfortably? Do uh, do they connect with the doctor electronic health record, which is the EHR? How did I do that? I used two main datasets. The first one was the mid dialogue. It's a collection of over like 500 real patient doctor conversation. I used a tool called natural language processing, which is the NLP, to find the most common health concern patient asked about. I also used a second data set, which was about like 100 plus mobile health app. I looked at the App Store rating, usability feature, and whether they followed like rule like the HIPAA, and the ADA Title II. I also checked if they worked with screen readers, like allow text resizing, and or if it have like a voice comment option. Also, last thing, I looked at how well these apps connected with electronic health record, the EHR system. I checked if they follow the HL7 FIRE or Smart on FIRE standards, which are the main way app and health system share information safely. This helped me understand how effective, usable, and accessible these apps really are. What I found with them from the mid dialogue, I saw the most common health concern, which was the COVID-19, chronic illnesses, and the pneumonia. This showed that patients use these apps for a serious medical concern, not just like a simple task. So these a lot of potential for these apps to use AI tools like a smart chat feature to give fast, helpful response. I also was looking at the app rating in future. Most app had good rating between four and five rating stars, but rating doesn't always show the full picture. Some highly rated apps were still hard to use or had missing feature like reminder or board notification. Apps that were simple, engaging, and personalized kept user coming back longer. Also, I was looking at the accessibility. A lot of app didn't work well for people with disability. Some didn't support screen reader. Other had tiny fonts or like a bad color contrast that make them hard or even impossible to use for people with vision or motor issues. Also, I was looking at the EHR integration. Many apps didn't connect well with doctor collection, electronic health record system. So even if patients track their symptom in the app, their doctor wouldn't see it. That's like a missed opportunity to make care better and like more connected with the doctor. I want to explain this first graph. This chart show how apps were rated in the App Store. Most fell between like 4 and 5 stars. Sorry, I'm looking at the wrong. This one. But again, like a good rating didn't always mean good design or accessibility. We need to look past the star and focus on like real potential. On the second chart here show the top 10 medical topics from Med-Dialogue conversation. Things like COVID-19, asthma, and chronic pain came up often. Apps need to be ready like to respond to these serious concern with like a smart, accurate, and easy understanding the information. Okay. And here to wrap it up, health app are helpful, especially for people managing long-term condition or like these who can't visit doctor easily. But many of them like still fall short for important areas like accessibility and usability, and also the EHR integration. To truly support the patient-centered care, we need apps that are like easy to use, accessible for everyone, and connected to the healthcare system. There is also a lot of potential to use AI to help answer common potential questions based on like real conversation, like the one I studied on in the mid dialogue. And also, this project was like to show me how important it is to design a digital tool that worked for everyone, especially in the healthcare. Thank you so much for listening. And here's some reference. And I would like to especially thank Dr. Isabel for going through this journey with me.
All right. Well, thank you very much. Are there any questions? Well, I do have a question. When you said that AI can help with the apps, can you be more detailed about how the generative AI can help with the apps?
Can you say it one more time?
Yeah. So you said that generative AI can be helpful for the apps. Can you be more precise? Explain more about that. How can generative AI be helpful with the apps?
So we all know how AI it's been like really helpful for everyone. Like they can like AI can read like for you if you can't like read, or they can like resize like if you want to like a specific size, if you have a specific font, they can change it. Like there's a way that we can make like an app that could be like more helpful and more easy for people who especially in need.
Mm-hmm. In terms of accessibility, is this would that be useful for accessibility, the
Yeah,
generative AI?
yes. That's the main reason I wanted to use AI.
Okay. All right. So these were my questions. Is there any other question? No? Well, thank you very much. Thank you for your presentation.
Thank you.
Okay, I will move on to the next person on the list. So we have, I'm just going to go back again to see if anybody's here. Okay, so Sravani, I see that you're here. I'm going to pin you. And if you could share your screen, I'm going to get a timer ready. Shravani?
Yeah, yes, I'm here.
Okay, I have the timer ready. You let me know whenever you're ready.
Hello, I'm Shravani Kota, and today my presentation is diabetes prediction using machine learning. So, diabetes is a rapidly growing health concern that can lead to severe complications if not detected early. Traditional diagnosis involves expensive and time consuming um, test. In this project, we explored how machine learning can be used as a faster, data driven alternative for early diabetes prediction using clinical health data. We built and evaluated various models to classify patients as diabetic or non diabetic. My aim and objectives. The aim of the objective project is to develop a robust machine learning model to predict diabetes based on patient health data, and our objectives include performing exploratory data analysis EDA, training and comparing multiple algorithms, identifying important predictors, and offering insights for improved diagnosis through data driven healthcare approaches. So coming to methods, we used a public data set from Microsoft GitHub with 15,000 records and numeric features such as body mass index, glucose, insulin, and age. The data was split into 70% as a test set and training as 30. And we performed pre-processing like normalization and checked for missing values. And EDA was used to identify trends, and we trained 6 models. So first one is logistic regression, KNN, and random forest, XGBoost, and artificial neural networks, and SVM. So hyperparameters were tuned using stratified six-fold crash validation. We used Python and skitlearn, XGBoost, and MLR for more model building. So coming to results, exploratory data analysis showed diabetic patients had higher plasma and glucose, BMI, and more pregnancies. So model performance was compared using accuracy and ROC accuracy. So in my project, KNN achieved 86% of accuracy, and random forest 89% of accuracy, and XGBoost 96% of overall accuracy. In neural networks, 87%. So XGBoost outperformed all the with others excellent sensitivity, specificity, and a curve 0.99. And feature importance showed that pregnancies, body mass index, and glucose were the strongest predicators. Coming to conclusion, this project shows the machine learning, especially ensemble methods like XGBoost and random forest, can reliably predict diabetes using health data. These approaches are fast, accurate, and offer support for early detection. The findings reinforce the use of predictive analytics in improving diagnosis and healthcare outcomes. Coming to future direction, future work can improve this system by addressing class imbalance using smart testing on more diverse and real-time data sets and incorporating wearable sensor data. We also plan to apply explainable AI techniques to make model outputs and more accuracy and interpretable to healthcare professionals. So in my project, I also include performance summary table. So this table shows the overall accuracy of each model and accuracy of each model, and XGBoost clearly performed best accuracy, followed by random forest. So neural network also did well, while simpler models like KNN and logistic regression had lower but acceptable accuracy. So in my project, I have like I tools used. We used Python for development, pandas and numpy for data handling, and XGBoost for modeling, and RStudio with MLR for benchmarking. Visualizations were created using Seaborn. My final statement is mission statement is our mission to use machine learning to make early diabetes detection more accessible, accurate, and personalized, supporting proactive care and improving long-term health outcomes. Thank you.
Okay, thank you. There was quite a bit of background noise. I don't know why, but okay.
Professor.
Yeah, can you explain to us what's your setup for training and testing? Because you do represent here training and testing, but
Yeah,
what's
as professor training, I take like two sets of data. One is training test, one is training data, and one is test data. So I,
how did you select your training and test data?
yeah, one is 70% and one is 30%. Because of high accuracy and compatibility and
Yeah,
precision accuracy curves.
yeah, and why did not you use cross-validation? Because it's stronger than using just a training and test set. Yeah, no, this is my question. Why did not you use cross-validation? And I would suggest using cross-validation because it's going to give stronger results.
As professor, I used cross validation for accuracies.
Oh, you did use
Is a
cross-validation?
technique used for machine learning and statistics to evaluate the performance of model accuracies, and it will generalize the independent data set because I'm taking from the GitHub.
So are you using cross-validation? Because it doesn't seem, when you show your pipeline, it doesn't seem that you're using cross-validation. So I was suggesting maybe using it.
I used cross validation and in test cases dataset. So I uploaded in the main file. So I use this because of like matrices.
Okay, well, thank you. Are there any other questions? No? All right, well, thank you.
Okay, I will move on to the next presenter. I'm going to close out the timer here. Timer information. Okay, so we have, please correct me if I say this wrong. Rukia,
Yeah, let me just, oh,
are you, it looks like you're next on the list. Do you think you are ready to present?
yeah,
Okay, all right, let me go ahead and pin you. And you can let me know whenever you're ready. Once you have shared it to the presentation mode.
okay, can you all see my screen?
Yes.
Awesome. Fine. So, my name is Rukia Sheikh. I'm a fourth year PhD student at SUNY Upstate in the Department of Biochemistry. And today I'm just going to give a brief overview on my current work, which is elucidating the interaction between interleukin-2 invisible T cell kinase, ITK, and its adaptor protein, SH2 domain containing leukocyte protein of 76 kilobase, or SLP76. So for background, we're just going to jump to figure 1. See if I can zoom in something. So just to give some disease context, when patients who undergo transplantation in the form of, or when cancer patients receive a transplant in the form of allogeneic hematopoietic stem cell transplantation or bone marrow transplants, these transplants have a number of cell populations that can have different effects. Can you guys see my mouse? Shoot.
Yes.
I'll get a pointer on here. Here we go. I don't know why I keep zooming back out. So these cells can have different effects. One is that they can, the matured T cells in this transplant can go and differentiate into different hematopoietic cells, or they can have the desired effect, which is targeting the cancerous tissue. This is known as graft versus tumor responses, or GVT. And there can be a third harmful response, which rather than targeting the cancerous tissue, the cells go and target the healthy tissue, and this can lead to things such as organ damage. And that response is known as graft versus host disease, or GVHD. So GVHD and GVT are normally coupled to each other. And what was shown by work done by our collaborators is that there's a specific interaction in T cells that can be targeted to uncouple the two reactions. So you get a reduction in the harmful GVHD responses while maintaining graft versus tumor responses. And that interaction is the ITK SH2. So the SH2 domain of ITK binding to the phosphorylated tyrosine 145 on SLP76. So in normal cases, when this tyrosine is phosphorylated, the SH2 domain will come and dock to tyrosine 145. This will lead to some downstream signaling and T cell responses. And then in the case of transplantation, it's what leads to this GVHD, GVT responses. But through targeting this interaction, through using peptide inhibitors or through knocking out ITK, like if you're doing cell models, you can get enough reduction of signaling downstream where you're reducing the harmful responses, such as decreasing the inflammatory cytokines, and increasing things such as migration of regulatory T cells, which helps increase the graft versus tumor response in lower GVHD. Now, this interaction has been studied to death in T cells and from immunology side, but we're interested in looking at what's going on from a molecular standpoint. But what's difficult about that is many of these proteins have been synthesized in vitro, but we do have some predictive structures to go off of to help us explain what's probably going on with the protein, or what's predicted to be going on with the protein. So here's just a prediction of what ITK looks like. And for this poster, I'm going to be focusing mostly on ITK. We have 4 major domains. Each of these domains have their individual functions. For today, we're just going to focus on the functions of the SH3, SH2, and kinase domain. So the SH3 domain, its main function is to bind to regions rich in proline residues. The SH2 binds to phosphorylated tyrosines, and the kinase domain is the center of the catalytic activity. And what's interesting is that through work done on similar kinases, they saw that this SH3, SH2 kinase domain, which is known as the circ-like module in other proteins, is highly important in regulating kinase activity. So we are predicting that it's going to have a similar effect in ITK, where when the individual domain ligands are introduced, they'll start to bind to their respective domains and open up the protein. This will then expose the kinase domain's active site, which contains a tyrosine that, when phosphorylated, increases the protein activity. So this can be phosphorylated either through autophosphorylation. So ITK molecules can go and they have some basal level activity, it can phosphorylate itself, or a different ITK molecule coming in and phosphorylating the protein in cells. So that's all for background. For our aims, we have 3 main aims. First is to look at the binding between the SH2 domain of ITK and different SLP76 based peptides. Then we're going to look at, or our next aim is to express and purify different kinase domain containing constructs of ITK, including that circ-like module that I just explained. And then the third is to look at the binding properties of this interaction in the context of our circ-like module or larger constructs. And to do this, we have a number of different constructs proposed. So we're taking two approaches kind of. We're looking at the interaction from the simplest form, which is the single SH2 domain and different SLP76 based peptides. And on a larger form, looking at the circ-like module and full-length ITK interaction with full-length SLP76. So for ITK, our constructs are mostly centered around the SH2 domain since that's our domain of interest. For SLP76, our constructs are centered around tyrosine 145, so they're building up from the individual tyrosine 145 peptides, which are shown here, going to the disordered region. So SLP76, the disordered proteins, that means it doesn't have a fixed structure, so it's kind of like wiggly like spaghetti. So it has this long region of disorder that contains different tyrosines that in cells is targeted by other kinases. But for our purposes, because we're looking at it in test tube, we need to consider the effect that these tyrosines might be playing on the ITK SLP76 interaction. So that's why we have them included here. But we have different versions where either the other tyrosines are mutated, so they're not phosphorylated, or we're including all the tyrosines, or we're putting the tyrosines in the context of the whole protein. So far, well, actually, here's my methods. So to do this, we're starting from transforming cells. We're using E. coli bacteria cells to transform, express, and purify our proteins. And then for the phosphorylations, we have the proteins, we're putting them into this phosphorylation buffer, adding ATP and different combinations of ITK with or without SLP76, either to activate itself or to test its activity on SLP76. And then this is analyzed through running gels. We have a standard Kumasi stain, which is just looking at protein expression levels, and then a stain that's specific for phosphorylated proteins. So the more phosphorylated the protein is, the more it'll uptake the stain. And then for binding, rather than dialyzing in the phosphorylation buffer, we're putting the protein into binding buffer and then using isothermal titration calorimetry, which is just a heat-based method to measure binding affinity. So I have here some of the data, just some of the representative data I've gotten so far for the binding. For ITK, the SH2 domain, we started to screen it against different peptides just to see which peptide binds stronger and if the results that we're getting in vitro agree with the in vivo data that our collaborators have gotten. And this peptide, the 145, 132 to 155, it was already tested in cells and was shown to reduce ITK signaling. So we're just kind of backing it up by showing that there's a strong interaction between the SH2 domain and the peptide in vitro, and they have a tight binding, which is shown through, we're getting just different binding curves, low KDs, basically, low dissociation constant. And then in terms of the phosphorylation, this gel is a lot, but just to summarize it, what we saw is that when we took our different kinase domain containing constructs of ITK, so the SH1 domain, which is just a kinase domain itself, or the circ-like module, we saw that both of them, when purified in bacteria, we were able to activate it. And that is mostly shown here in this lane that's brightly staining. So here we just have a different combination of ITK, the circ-like module with the SH1 domain and the SLP76. And the high uptake of the stain shows that our protein is active and it can phosphorylate its substrate. So our current conclusions include that we were able to show that there's in vitro binding between the SH2 and different Y145 peptides, and that there's a preference for this 132 to 155 peptide, which was already tested in cells. And then additionally, we have an active circ-like module as well as active SH1 domain, and now we can start to incorporate those into our binding experiments and start to look at how this complex is forming as a whole in vitro. So for our future studies, we want to go forward with our binding studies to move up from our SH2 and peptides to our full-length proteins. That's it.
Well, thank you very
Yeah.
much. I don't know if there is any question from the audience. Well, you can raise your hand if you want. I had a question myself because I see that you have shown the AlphaFold, which is kind of a 3D protein structure prediction. So it's very interesting. So maybe some in the audience may not know what is AlphaFold. I know it was the Nobel Prize last year, 2024. I mean, in 2024. And so the question is, can you explain what is AlphaFold, how you are using it? And then the next question will be about how do you see AI and generative AI contributing to your work?
Yeah, I mean, so AlphaFold, as far as my understanding, is just like a protein structure predictor. So it looks at the protein sequence and compares it to other proteins that are similar and kind of generates this overall predicted structure. So, like I said, there's no full-length structure for ITK, but there are similar proteins that come from the same family of ITK that have been studied and their structures have been found. So it'll take the sequence of ITK and compare it with those structures and those sequences and generate a predicted model of what the protein's expected to look like, not what it does look like. So that's what's important to keep in mind is that all of these are still predictions. There's no 100% definitive, this is what the structure looks like. Sorry, and what was the second question?
Yeah, the second
Sorry.
question was
and how am I using AlphaFold?
yeah, how do you see
Yeah.
that generative AI contributing to your research in the future? The
I
kind
mean,
of
like
research you're
AI
doing.
is huge for this type of project because I'm like because I don't have an actual protein structure, I'm highly dependent on these predictive models for what my protein would look like. As well as because it's a disordered protein, we're looking at it because it has some regions of disorder, not it's a disordered protein. It has some regions of disorder. We're using AI to estimate where these disordered regions are and how they could be contributing to the protein's function. And additionally, because we're looking at binding, there's a number of different predictive platforms that we can use to help us kind of narrow down what experiments we need to do on the bench by using those predictions to determine what are the best peptides or what are the best conditions for us to test in vitro. I mean, like on the bench. So
Yep.
there's a lot of applications for AI in this project.
Excellent. Well, very interesting. Any other question? No? Okay, well, thank you very much. Very interesting.
Thank you.
Okay, next we have Yu Ping. I will pin you right now. If you could please share your screen. And I have the timer ready whenever you're ready.
Can you hear me?
Yes, I can hear you. Do you mind expanding it so that it's like in presentation view?
Okay.
Yes, works. All right, let me know whenever you're ready.
Okay. I think I'm ready.
All right, go ahead. The timer's already started.
Okay. So hi. I'm Yuping Zhang. I'm from SUNY Oswego, and my topic is analysis of the impact of obesity and poverty on diabetics using machine learning methods. So diabetes is a very common chronic disease caused by the abnormal accumulation of blood sugar in the bloodstream. And this study will focus on the impact of obesity and poverty on diabetics in the United States. The data on obesity, poverty, and diabetics are obtained from the United States Diabetes Surveillance System and the U.S. Department of Agriculture. And then the data will combine and we conduct the analysis on the data. We perform correlation analysis, association rule mining, regression analysis, and classification analysis for this study. So just little more details about diabetics. It's a very common chronic disease caused by the abnormal accumulation of blood sugar in the bloodstream. and it occurs when the pancreas does not produce enough insulin or when body cells become resistant to insulin. In the United States, there are more than 38 million people have diabetics, approximately 1 in 10 individuals factor for diabetics and can be considered as a biological factor that directly involved in the mechanism of diabetics development. Many studies have found that the accumulation of adipose tissue can contribute to the overproduction of insulin by the pancreas and induce insulin resistance, which can lead to diabetics. Poverty is a potential risk factor related to diabetics and can be considered and according to the CDC, individuals who with a family income about 500% of the federal poverty level have the lowest prevalence of diabetics. And the prevalence increases as income levels decrease. Those with a family income below 100% of federal poverty level experience the highest prevalence of diabetics. The Pearson correlation coefficient was calculated to measure the strength and direction of the calculates to assess the statistical significance of the correlation. We generate scatter plots for obesity, diabetics, as well as poverty and diabetics. And for the association rule mining, we use the a priori algorithm. And for regression analysis, we use linear regression support vector machine. And for classification analysis, we use decision tree, knife bay, SVM, KNA, and then random forest and logic regression. I have to access the full screen. I think PDF, I cannot expand the sessions. Okay. So here is a correlation analysis result. So we could see there's a pretty strong correlation between obesity and diabetics, and moderate positive correlation between poverty and diabetics. The p-values are below 0.05, indicate there's statistical significance between the variables, and we could also see the positive correlation on the scatter plot. And this part is the result from association rule mining. And these are the top 4 frequent associated items. So we could see that high obesity are frequently associated with high diabetics. Low obesity are frequently associated with low diabetics, and same thing between poverty and diabetics. And the leaf value are above 1, indicates the positive association between the items. So here is the result of the regression analysis. We could see that the correlation efficient is about 0.6 for when we use obesity alone as a predictor, with 0.64 as the highest. And for the when we use poverty alone as a predictor, the correlation coefficient is about 0.68. And when we combine them, the correlation coefficient is about 0.72, 73, and with 0.75 as the highest. And move on to the classification. So for classification, we use 7 models. And when we use obesity alone, the accuracy for most models is about 74%. And for poverty alone, it's about 66%. And when we combine both predictors, for most model accuracy, it's about 78%, and the highest is 78.85%. And these are the confusion matrix. This is the confusion matrix of naive Bayes, and this is for logic regression. We could see that the model is doing pretty good on predicting the low diabetics and the medium diabetics, but they are kind of struggle with the high diabetics. And when we use both obesity and poverty as the predictors, there is improvement in predicting the high diabetics. But we could see that the instance for high diabetics is only 31. So that's probably why the models were struggled. That there are not enough data for them to study from. So in conclusion, sorry. So the correlation analysis of obesity confirmed that obesity has a strong correlation with diabetics. And the poverty has a moderate association between with diabetics. And association rule mining validates these relationships by identifying the patterns within the data. And high rates of obesity and poverty were associated with high rates of diabetics. And low rates of obesity and poverty were also associated with low rates of diabetics. And machine learning methods involving regression and classification demonstrate that obesity and poverty are effective predictors of diabetics with their impact qualified through model accuracy. By integrating both obesity and poverty into regression and classification models, the accuracy of diabetics prediction was enhanced. This approach highlights the importance of accounting for socioeconomic factors with biological factors in diabetic research, and it's particularly relevant when applying machine learning technique to diabetic prevention. Okay, that's it.
Right.
Thank you.
Well, thank you for your presentation. So my question is, what further analysis do you think would be interesting? I mean, you have any remaining questions following your work, and also what would be next to explore?
As I talk about the confusion matrix, I think the high obesity level for the classification is too little. So I think when we build the model, it's better to disgrace the samples like to make the high instance more. So we'll have more data for the machine to study from.
Mm-hmm. Okay, that's a good suggestion. Very good. Is there any other question from the audience? No? Well, well, thank you very much. And I think this was our last presentation for this poster session. I see somebody joined in, but I don't think he's a presenter. Or do we have any other presenter who was not presented? Or
No, that it
no?
looks like everybody is. Some people are joining in from other groups because they've also finished.
Okay.
Yeah. So, but we are done here. So
Okay.
you can all leave the room. If you leave the room in the bottom right-hand corner, it'll send you back to the main room, and then we can just wait there for closing.
Okay. Thank you so much. Thank you, everyone, for your
Yes, have a good one.
presentations.
Thank you, Professor.
Thank you.
Thank you, Professor.
Thank you.